when we talk about blood, there's a lot of people are under the assumption that blood is just a liquid, and it's not. There are 4 components of blood. The liquid component is plasma, so you've probably heard the term plasma before. But then we also have 3 formed elements. Formed elements are the actual blood cells. So you have red blood cells, white blood cells, and platelets. And once we get into these notes, you'll see that red blood cells are called erythrocytes, so you always see that. White blood cells are always called leukocytes, and I'm going to make some distinguishing comments when we get to leukocytes. And platelets are called thrombocytes. So when it's referencing formed elements, it's red, white, and platelets. Blood total, the total blood composition is plasma, red blood cells, white blood cells, platelets. Okay? So when you talk about blood, it's not just a fluid, it's plasma and formed elements. There are 4 aspects, or 4 components of blood. So I mentioned erythro, leuko, and platelets. A lot of individuals, and we'll come back to this when we get into leukocytes, but you've heard of lymphocytes, and then there's leukocytes. So leukocytes means it's a white blood cell. Lymphocytes is a type of white blood cell. And I'm going to show you on a chart. Okay, so make sure when you're reading questions, you distinguish between leuko and lympho. Okay? And what we're talking about, or what you're being asked there. So plasma, the formed elements. Whenever you, if you've ever heard of hematocrit, and those of you who have worked in an office or you are phlebotomists, we take your blood and we put it in a centrifuge. And a centrifuge is it may look like this. There are different types of centrifuges. But you put it into a test tube, then you cap it off, and you put it into a centrifuge, and it spins it very quickly. And when you spin it very quickly, it separates the components of blood. It doesn't keep it as that whole mixture. It begins to settle out. And we call that the hematocrit when we just get your red blood cells. The red blood cell count is your hematocrit levels. Okay? This area right here is your white blood cells. Okay? And then here we have our plasma, our condensed fluid portion of our blood. Let me go back and make sure I've covered everything. Yes. And then hematocrit, yes. Okay, textbook numbers. I don't know if I've said this in O2, so I just want to say this real quick. All the numbers that are offered here are all textbook averages that were present at the time that the material was printed. So it says that the percent hematocrit levels for men or males should be higher than females, and we would expect that on average. But could you have a patient that's a male have a lower hematocrit level than a female patient? The answer is yes. So these give you kind of a range. In general, men have a higher hematocrit level than females. That doesn't mean that it's always the case, but just generally speaking, that's the situation. So 47 plus or minus 5% of the blood composition for males should be red blood cells, and 42, which is a little bit lower for females. And you'll hear me reference textbook numbers quite a few times. When we look at the physical characteristics of blood, if you've ever felt blood before, it's not like water. It's kind of a stickiness to it, and if you let blood dry, it actually kind of sticks to you. And it crusts up, even when it does dry out. It's not like water, where it just completely evaporates. There's formed aspects of it, the elements. So it's kind of this sticky, viscous, kind of thick fluid. It is a dark color. If it is low in oxygen, so low O2 levels, we have darker blood. If it has a light or a high level of oxygen in it, it's a bright red color. I often get asked, so your blood is blue in your body until it touches air? And the answer is no, never. Your blood is never blue. Your vessels appear to be cyanotic, which we discussed in the integumentary system. We looked at the coloring. Cyanotic just means that that aspect is low in oxygen. But your blood was never blue. The vessels were low in oxygen, so they appeared a cyan color, a cyanotic color, so a bluish green. But your blood itself is always red. It's either dark red or light red, depending on whether it's got oxygen or not. So a lot of people will say, oh, it's blue blood. No, it's not. Okay, so you only have red blood. Just depends on what shade of red, whether or not your oxygen levels are high or low. The pH of your blood should be around 7.3, 7.4, 7.5, 7.2. So there's a range. Whatever your homeostatic range is, we want to keep it in that range because too acidic is lethal and too basic is lethal. And we've discussed previously, but buffers. So if your blood becomes too acidic, immediately your body tries to maintain homeostasis and will throw base in there. And if it becomes too basic, it'll immediately start to throw acids in there to try to maintain homeostasis. Your blood and pH is something that we don't mess with. It becomes lethal very quickly. And generally speaking, it's around 8% of your body weight in the average volume of males versus females. Of course, it's mentioning males having a little bit more blood than females on average. What's the function of blood? It distributes substances, so it travels everywhere throughout your blood. Main substance that it will distribute is going to be oxygen and nutrients. Okay, it's responsible for regulating blood levels of substances like calcium, glucose, helping us to maintain homeostasis. And then the last one is for protective purposes, and that's going to get us into the platelets and the leukocytes, immunity, and clotting. Okay, so the distribution functions of blood include delivering oxygen to all of your tissue. Whenever something, and this has come up previously in lectures, it says that it's avascular. Avascular means that there is no blood there. If there's no blood there, that means that there's no oxygen there. If there's no oxygen there, that tissue is dead, or it is not a thriving tissue that can grow and repair itself. In any of your tissues right now that do currently receive blood, if we were to cut out the blood to that tissue, what would happen to it? It would die. And depending on where that tissue is located would determine whether or not it would regenerate. If the oxygen levels were low in your heart and you had a heart attack, that tissue doesn't fix itself. And we can see scars of that. We're going to talk about that in the next lecture. But in your skin, if you cut your skin and the blood vessel ruptures, will it eventually repair itself? The answer is yes, because it's an epithelial tissue. So when we talk about avascular, it doesn't have blood, so it doesn't have oxygen. Oxygen is necessary to produce ATP, which is energy, right. So if that tissue needs oxygen, it's because it needs energy in order to carry out its metabolic processes. And remember, metabolism is building up and breaking down. It's not just like digesting food. Okay? So there's a huge function for oxygen throughout our body. It mentions also that the blood is responsible for transporting metabolic waste. So as we are traveling through our body, or as our blood is traveling through our body, we have cells that pick up things that aren't supposed to be there anymore. Whether it's damaged cells that have been working for us but have expired, or antigens, things that don't belong there anymore, like dirt, or maybe a bacteria or something that got in. It picks it all up and it helps us get rid of it. Okay, and we're going to learn that in our lymphatic chapter as well. If it's going to be eliminated through our urine, it'll go to our kidneys. If it's going to be eliminated through our feces, it'll go to our feces, which is your... Okay, transporting hormones, and we discussed hormones in the endocrine section in the last lecture, but if it's endocrine, it's released into the blood. And so when that hormone is released, it goes anywhere the blood goes. And the only organs that respond to it are organs that have the receptors for that hormone. We discussed that. Regulation functions, this is specifically referencing temperature and pH. The two things that can cause the damage the quickest, temperature and pH. And you're going to see that theme in all living organisms, temperature and pH. If you get too hot, you'll denature. You'll change its shape, so you change its function. If you get too cold, you'll slow the metabolic processes down to where it can't maintain life. If the pH changes, whether it's acidic or basic, it will also denature. It'll change its shape, so it will change its function. Temperature and pH are the two main focuses of homeostasis. Okay, and of course it mentions here that blood is responsible for regulating both. If you're too cold, your blood is shunned into your vital organs, and you start to shiver in order to help bring up your body temperature. And if you're really hot, your blood flow increases, your skin starts to turn a pinkish reddish color, and it's helping you to get rid of heat. That's the purpose of that coloring in your skin. As far as maintaining pH, it uses buffers in order to keep, if you're too acidic, it gives you bicarbonate buffers, and if you're too basic, then it gives you acidic buffers in order to maintain your blood pH. Protection functions. When you're talking about blood as a protective mechanism, there's platelets. Platelets are responsible for blood clots, so if you have some type of laceration, cut, so some type of compromise in your tissue, platelets are supposed to coagulate and create a plug so that you stop losing blood. That's a protective function, so that you maintain your blood volume. The second protective function is carried out by leukocytes, which are your white blood cells. And your white blood cells carry out what we call our immunity or our immune function. And we'll get into that again. There's a whole chapter on that. So we have our antibodies. We'll discuss complement proteins. And white blood cells, you have 5 types of white blood cells. So that's why I'll say when you get there, I'll distinguish between all of those. Any questions so far? So we went through the functions of blood, kind of the characteristics of blood. And now we're going to go through each of the components of blood, beginning with plasma and then going through each of the formed elements. So the blood plasma is the liquid portion of your blood, and it's mostly water, surprise, surprise, which is why water is such a huge part of our composition, personally, individually. We have to keep our water levels high, maintain hydration in order to keep our blood volume homeostatic. Whenever you talk about what's in your plasma composition is probably a little bit different than my blood plasma composition because we live different lifestyles, and do we eat the exact same thing? No. And do we have the same nutrient requirements? No. So, although these are it could be a number of these things, and it might not be one of these things. So it mentions that you might, well, of course, hopefully you have nutrients. But gases, it's specifically talking about like oxygen, carbon dioxide. It's not talking about that you have like methane or anything like that inside of you. That's not what we're getting at. Hormones, waste, proteins, inorganic ions. Inorganic ions are whenever you say vitamins and minerals are inorganic. Okay, so that's what it's referencing there. You also may have some plasma proteins, but you should have. The amount of plasma proteins may vary. But these are proteins that you find in your plasma, which is why they're called plasma proteins. Yeah, that's why they're called that. There are 3 classes of plasma proteins: the albumins, the globulins, and the fibrinogens. Okay, and it gives you kind of a rough estimate of how much of each of those plasma proteins arrange for that. Now, this chart I included because it explains every component of the blood, the 4 components of blood, and it gives you details for each. So it's talking about here, this is the plasma. Here are the components that you will find in plasma. So electrolytes, and when you say electrolytes, this is what we mean by electrolytes. All of these inorganic ions. Okay? Or it could be, there could be some ions there. Plasma proteins, albumin, globulins, and it's giving you the percentages again. Then you have fibrinogen, okay, and then it gets into your respiratory gases, hormones, anything that could be possibly found in plasma is listed here. And again, individually that might differ for a person, but this is a catch-all for that. Okay, so that was plasma. Any questions on plasma? Okay, the majority of the rest of this chapter is red blood cells because they have a really important function, obviously. But then we go short into white, short into platelets, and then we'll get into developmental aspects. So the formed elements, we have the white blood cells, which are going to be abbreviated WBC. We have the red blood cells, which are going to be abbreviated RBC. And we have platelets that aren't abbreviated. They're just called platelets. Okay? It mentions that white blood cells are complete cells, which means that they have a nucleus and they have all of the cellular organelles that we studied in O1. So like your Golgi apparatus, your endoplasmic reticulum, all of that stuff you're going to find in white blood cells. And a nucleus, which is the big deal. Red blood cells are, when they're mature, they have no organelles because they're designed to carry oxygen. So when you take out a red blood cell from someone, it doesn't have a nucleus that has their genetic material stored in it. Okay? The cells around it may, like if there's white blood cells, that might have genetic material, but red blood cells purge all of their organelles right before they become mature and start working. Okay, so it mentions that they have no nuclei. They're what we'll end up calling anucleate. Platelets are cell fragments, and we'll discuss how those are formed as well. Okay? Many of these cells, formed elements, red, white, and platelets, only survive for a few days. And then once they've carried out their function, they're destroyed, and we pee them out. Or you poop them out, which is what gives your feces that color, that reddish, brownish color. And that's because of all the red blood cells that you're getting rid of. Red blood cells have the longest longevity. They can live the longest, but they're actually not living because they're not dividing or anything like that. Once they're done, and they just work for about 3 months. White blood cells, they have a shorter lifespan, especially if you have an infection. So they're trying to cycle themselves out. And platelets, we're talking about like 2 weeks at max, and you're getting rid of those platelets. All of your blood cells, so red, white, and platelets, are all going to be manufactured in your bone marrow. Okay, so right now as adults, the majority of our red blood cells, any blood cells, are made in our femurs. This is 100%. Now, you still make it here in your sternum and in your distal epiphysis, or bones of your humerus and so on. You make it other places, but the large portion of it is made here in your femurs, which is why femoral injuries are lethal. And you could be riding your bicycle, crack your femur, and there's a good chance they're going to care flight you somewhere, because that's where your blood is made. And you could do the same thing and break your arm, and they're not going to care flight you. But it's because of the femurs. The femurs are a big deal as far as blood cells are concerned. Okay? So we'll mention that as we get a little bit further. Here is a slide of blood, and if you were with me last semester, we talked about blood is one of the most difficult things to focus, and when you can focus red blood cells on a microscope, you're a beast. Okay, so here, these are all red blood cells here, and I can tell because there's no nucleus, there's no colored portion of it, so they're just a cell carrying oxygen. These have nuclei. This is not a full cell. Those are platelets. You can tell that that's all kind of broken down. But these have nuclei. These are white blood cells. And they're different types of white blood cells. You can see they all look a little bit different. But the majority of your blood composition is going to be red blood cells. Okay, so erythrocytes, which are red blood cells. Erythrocytes, when we just talk about their general characteristics, what they look like, they're biconcave. So last week when we talked about convex, when we looked at our lens, we talked about it being like this. Red blood cells are biconcave. When you look at them, they look like a donut that doesn't have the center punched out. So just a donut that's just kind of flattened there in the middle. It also mentions here that they're anucleate, which I discussed previously, but again, they do not have a nucleus when they're mature. They have no other organelles. They purge all of those right before they become mature, and they're carrying out their duties. They are crazy small, but their design is intricate. They are exactly the diameter of one of your smallest blood vessels, which are your capillaries. So if you watch, you can actually YouTube this, because I've done it before too. You can see that red blood cells will go through your capillaries in a single file line, because they can't go any further. They're exactly the diameter of your smallest blood vessel, your capillaries. And that's where they can drop off oxygen and pick up carbon dioxide. And that's their whole job. So it works out perfect. They are the exact size and shape to efficiently carry out their function. They are filled with this protein. This protein is hemoglobin, and it's going to be abbreviated capital H, lowercase b, pretty much for the majority of the notes. But hemoglobin is a protein that will allow for the attachment of iron, but also the attachment of oxygen. So hemoglobin levels, we're trying to figure out if we can grab onto oxygen or not. Okay, it mentions that the protein spectrin is also there in the plasma membrane of a red blood cell, and it tells you that spectrin, this protein, provides flexibility for that red blood cell to change shape. The reality is that if we stayed in the same position all the time, that this wouldn't be a big deal. But we bend over and tie our shoes, so we kind of compress some of our blood vessels momentarily, and we don't want a blood clot to start there immediately. Okay, and when you go to sleep, same thing. So they can temporarily change shape as they're kind of squeezing their way through your vessels, but then they kind of pop back out and do what they're supposed to be doing. Okay, and that means in the event that there's no injury, like this is just your normal everyday activity. The red blood cells are what contribute the most. They contribute the most to your blood viscosity. And viscosity, again, is the thickness of your blood. Okay, a low viscosity means that it's very runny, kind of like water. A high viscosity means it's like syrup, like a thick syrup. Okay, so here's kind of what a red blood cell should look like. It mentions still the structural characteristics, that it's good for carrying gas. It's a biconcave disc. It has hemoglobin. There are no mitochondria. Mitochondria are responsible for producing ATP, which is the energy, because remember, red blood cells don't have any organelles once they mature. So they go through anaerobic respiration, which means that they produce a whole lot less ATP, but that's okay because you have billions and billions of red blood cells carrying out this process. Superb example of a complementary structure and function. These red blood cells are specifically designed to carry oxygen and fit through the smallest capillaries, which is where gas exchange occurs, oxygen and carbon dioxide, and they carry this. They do it perfect. If you're healthy. Erythrocyte function. It still mentions here that their main responsibility is their transportation of respiratory gases. You have two respiratory gases, oxygen and carbon dioxide. Okay? It mentions that hemoglobin, which is the protein that we find inside of red blood cells, binds reversibly to oxygen. And let me explain to you what that means. That means that when it initially binds to oxygen, so like I'm inhaling right now, my red blood cells that are in the capillaries within my lungs are grabbing onto the oxygen molecules that I have inhaled or inspired. Then they will be transported through the rest of my tissue. And wherever they are supposed to drop that oxygen off, like if it is my big toe, when it gets to my big toe, the hemoglobin will let go of that oxygen so that it can serve that tissue. Does that make sense? So it holds onto it, and then it lets it go at its specific destination. When it says it binds reversibly, that means it can hold on to it and then let it go, because that's its job. All right, it doesn't permanently bind to oxygen. It also mentions here the red blood cell count if we're talking about males versus females, and this is not hematocrit levels. This is giving you a different measurement tool. Okay, so hematocrit was when we talked about just separating out the red blood cells. Okay. This is actually the protein hemoglobin, and this is not randomly shaped because we know the only thing random in biology, so the study of life, are mutations. But here's an iron, so it mentions iron, and we'll talk about anemia here in a little bit, so I want to make sure that you see that. So here in this hemoglobin molecule, we have iron, and hemoglobin is responsible for binding to oxygen chains, or oxygen gas. I said chains, I think it's because I just saw globulin change. Some terms you need to be familiar with. When that hemoglobin molecule has an oxygen on it, we call it oxyhemoglobin. So it's got oxygen on it, and it's hemoglobin, so it's oxyhemoglobin. When it lets that oxygen go, we call it deoxyhemoglobin, because it let it go. Does that make sense? And then if it picks up a carbon dioxide, which we hope it does to help get carbon dioxide out of our tissues, we call it carbaminohemoglobin. So oxyhemoglobin means it's picked up oxygen, deoxy means that it's let it go, and carbamino means that it's picked up the carbon dioxide. And where is our red blood cell going to take the carbon dioxide? Exactly, right back up to our lungs, where we're going to do what with it? We're going to exhale it. Right. Okay? So, and we'll talk about that process as we continue to make it through this chapter. So it also compares the coloration of blood as well. Hematopoiesis. So any real questions, any fake questions on erythrocytes? Because now we're going to go through hematopoiesis. Hematopoiesis is the general making of blood cells, and then we're going to specifically look at erythropoiesis which is specifically erythrocytes. I do want you to understand and distinguish those terms, because here it says hematopoiesis. Hematopoiesis is just the formation of blood cells. Hematopoiesis is just the formation of blood cells. It does not distinguish between what type of blood cell at this point. So hematopoiesis. In adults, where does this process take place? Wherever red bone marrow is. So axial skeleton, the girdles, proximal epiphyses, and of the humerus and the femur. So that's, and again, a major blood portal for us is going to be our femurs. Okay? Hematopoietic stem cells, or hematopoietic stem cells. These are stem cells that can become blood cells. There are 3 types of blood cells, so don't forget that. So you're going to see hematopoietic still. Hematopoietic anything, hematopoiesis, or all blood cells. So that could be red, white, or platelets. Until it specifically tells you this is what we're making, you cannot make any general assumptions. All blood cells start off with the same stem cell, whether you're making red, white, or thrombocytes, so your platelets. Okay? Then it will determine, hey, I'm going to become a red blood cell. Once it decides to become a red blood cell, it can't say, wait a second, no, I want to become a white blood cell. It mentions here that once they've committed to a cell line, they cannot change. They all start out the same, but once you commit to one, you can't back up and say, oh wait, I want to be a platelet. Okay? So they're forced to complete their life based on whichever way they commit to. Once that blood cell, any of those blood cells are ready for circulation, they'll enter the sinusoids and go into circulation. Okay, so right here, we're talking about erythropoiesis. So now I've specifically given details and said I'm going right into red blood cells. Hematopoiesis is all. Erythropoiesis is red. Leukopoiesis is white. And then we'll end up calling platelets thrombopoiesis. Okay? So you can distinguish between those. So now I'm talking just on red blood cells. And this gives you the stages in words. I like to use the diagram, so here's the diagram of it. We start off with the stem cell. And we go through a differentiation process. This is the maturation process of a red blood cell. And it's showing you, at this point, it's committed to becoming a red blood cell. And it continues to go through the divisions. And at this point, look at what it's doing. It's kicking out its organelles. And specifically here, we have it diagramming the nucleus. But it's purging its organelles. And it goes from that to looking like this once it's mature. Okay? So here are the stages of erythropoiesis, the formation of red blood cells. Okay? Just kind of going through those stages. Do we want this to take place quickly? No. We want this to take place in a calculated manner so that this is designed and shaped exactly the way it needs to be to successfully carry out its function. When you have red blood cells or any cell, and we discussed this in 01, that is made too fast, what do we call that? What disease is that? It usually is a result of a mutation, but anytime you have cells that are being created way too fast, it's cancers. Okay? Anytime cells are not taking their time to be produced, it's a cancer. If they're speeding it up. And a lot of times, whenever you were little, they were like, if you did it right the first time, you wouldn't have to go back and do it again, right? That's cancers for you. Cancers are doing it so fast, they're making mistakes, which is why it becomes a problem to you and I. Okay, so we do want this to take its time. How do we regulate erythropoiesis? How do we regulate the formation of red blood cells? We use hormones. Okay, and we're going to discuss that here in just a moment, an EPO. But if you have, we want the homeostatic level. If you have not enough red blood cells, then you don't have enough oxygen, so you're hypoxic. And what's hypoxic? Your oxygen levels are low, right? And if you have too many red blood cells, that becomes a problem because your blood's way too thick. It's too viscous. Okay, so that becomes an issue. We are making around 2 million red blood cells per second. But that's okay because we're getting rid of just that many, right? The whole reason we're producing cells is because we're losing them at the same-ish rate. We want to keep that balance homeostatic. We have to do that by regulating hormones and our diet, making sure that we're taking care of our body. So as long as you're healthy, your red blood cell count should be pretty homeostatic. We're going to mention hormonal controls and then making sure that we're getting everything that we need from our diet to make red blood cells. The hormone that's responsible for creating red blood cells, erythropoiesis, is regulated by the hormone erythropoietin, which is going to be abbreviated EPO. And you may have heard of EPO before. We hear this on the news a lot with athletes and blood doping, and I'll get there. But EPO is the hormone that is responsible for stimulating and regulating erythropoiesis. It is produced by your kidneys, directly by your kidneys. This was mentioned at the very end of our hormone chapter previously. Okay? So we should always have small levels of EPO in our blood, meaning that we're constantly telling ourselves to make red blood cells because we're constantly breaking them down, and that's helping us to maintain homeostasis. It is released by the kidneys, and in extreme cases it could also be released by the liver in response to hypoxia. So if your oxygen levels get really low, not just for a quick second, like if you hold your breath, it doesn't immediately cause you to make new red blood cells. But if your oxygen levels become low because there's an issue, whether it's medical or perhaps you're going on vacation in Colorado, somewhere where the altitude is higher, your blood cells say, wait a second, we don't have enough oxygen, so immediately you begin to release more EPO. And when you release more EPO, guess what you make more of? Red blood cells. Because they have a higher affinity to oxygen, they're going to try to grasp on to as much oxygen as possible. And, well, I don't think we talk about it today. I think we wait till the respiratory chapter. But if you've ever gone on vacation in a higher altitude, it's not uncommon for the first day or so for you to have to become acclimated. So you might get a headache or feel nauseated. And a lot of times those people go to sleep because that's their only way of handling it. But it's their body has to physically catch up, and it takes 2 or 3 days to do that. So if your vacation's 5 days long, the first 2 or 3 days you're acclimating. Day 4 is good, and then day 5, you're on your way out. But you'll be good for when you get back to the lower elevation. It could, oxygen, the fact that you have a low oxygen could be one of many things. Okay, yes, for sure. But I just don't want you to think it's because you're holding your breath like underwater or something. You don't immediately start making red blood cells. Yes.
Does it take like that long to, when you go back to the normal?
For your red blood cell levels to go back down? Yes. That's a great question. And sometimes a little bit longer because it's waiting to make sure that you're good. Yes, good question. What are some causes of hypoxia? So these are just generally speaking, but we can have a decreased number of red blood cells. So for some reason your red blood cell count number is going low. This is usually because you're injured. It could be that you, and it's going to use stab wounds. It uses stab wounds for every injury here. So perhaps you got shanked and you have this like bleeding stab wound or a gunshot wound. Your red blood cell count is going down, so oxygen levels are going down. That makes sense. You might also have like this slow little leak, like an internal bleeding or like a hemorrhoid or something like that, where you're losing blood slowly. We also see red blood cell loss there. Okay? You might also have an inefficient amount of hemoglobin. This is usually due to iron deficiencies. Okay, so whenever someone says I'm anemic, this is what they're referencing, and I'll get there in just a moment. And it might also be that you don't have enough, you're hypoxic because there's not enough oxygen in the atmosphere, so that's what we were just discussing on the acclimatization. Effects of EPO. When erythropoietin is released, it tells hematopoietic stem cells, I need red blood cells. So those hematopoietic stem cells say, okay, I'll become erythrocytes. So it stimulates them to commit to a bloodline. So it says, I'll become a red blood cell. It mentions here that some athletes will abuse artificial EPO. You can buy this on the black market, whatever. But what this does is if you inject yourself with EPO, it tells your blood I need more red blood cells. You can increase the number of red blood cells. You can increase the amount of oxygen that's in your tissue, and that increases your endurance. So we see a lot of endurance athletes doing that. They'll inject themselves with artificial EPO, and it'll allow them to run further, longer, or bike further. And it's endurance activities where oxygen levels need to be higher. Okay, so we see that. Testosterone naturally increases the release of EPO, so males naturally have a higher level of red blood cells than females, and we've looked at those textbook numbers. Okay, here is our homeostatic balance. This is negative feedback, so if red blood cell count levels are low, we're going to release EPO and make red blood cells. If red blood cell counts are super high, we're going to stop releasing EPO until we reach homeostasis. Let me chime in one more time on the EPO, synthetic EPO. You inject yourself with EPO, your body stops releasing it because the levels are so high, right? So you have to kind of keep doing that while you're doing those activities. So we see this happening right before performance. So like 2 or 3 days before performance, and we saw this in Olympic athletes where they were using artificial EPO. We also saw them using growth hormones, but they were using equine growth hormone. That's crazy, but that's why their muscles were so solid and huge, especially the track runners. But that's also why they test you multiple times before your competition and after your competition to make sure that you haven't you've played by the rules. So as far as what you need to be eating to make sure that your erythropoiesis or your red blood cell formation is at where it needs to be, you need to have a balanced diet. It mentions here nutrients, you need amino acids. Do you remember what amino acids are from? Proteins, lipids, fats, and then it mentions carbohydrates, which are your sugars. So this is your normal, you're eating. Now, you're eating well, so you're getting a nice balance of all of this. You're also going to need iron. Now, not to just go eat iron on its own, but you should be eating foods that are high in iron, like if you're eating vegetables, and those minerals are going to be found in healthy foods. B12 is responsible for your ability to absorb this. So you're going to see people with issues, and we're going to talk about this in a little bit, who have low levels of B12. They're anemic, and it's because they don't have the ability to absorb the nutrients from their food. B12 is really important. Okay, if you're just taking a multivitamin, you get your B12. I'm telling you, take a multivitamin and it will save your life in the long run. Just do it. You'll get what you need. If you don't want to swallow pills, Flintstone it. 2 of them because you're an adult. Okay, but get what you need because you will forego problems when you're old. Yeah, okay, see? Mark my word, I'm telling you, it has everything you need to carry out your normal metabolic processes. It also mentions folic acid. And if you're a female and your reproductive years, you should be taking folic acid anyway. And because that has an issue, or that leads to brain development in your offspring. So you want to make sure that there's plenty of that, and if there's a chance that you could become pregnant. As far as lifespan for erythrocytes, 100 to 120 days. So here you have like 3 to 4 months. Here's a range. Again, that's just an average. But remember, they don't carry out protein synthesis. They don't grow. They don't divide because they have no organelles. Their full job is to transport oxygen to and from tissue. Oxygen and carbon dioxide, gas exchange. What happens when a red blood cell becomes old? It becomes brittle and it breaks down and you filter it out. Your spleen will filter it out and eventually it'll become part of your feces and you'll poop it out. So that's what happens. That's what happens to your red blood cell, the life of a red blood cell. And it kind of depicts that here. Goes through and whoop, there you go. Feces excreted in the feces.
So Bruce, when you had your spleen removed, how would that
That's a great question. So when you have a splenectomy, because it's not a vital organ, other organs can pick up and carry out that function of that spleen. So you have collateral organs that will do the same thing. Yes, ma'am. Oh, for sure. Oh, for sure. Yeah, so a lot of times when they say they want a stool sample, they're looking at the color the consistency, any contents that that may or may not be there that they're looking for, or something extra that shouldn't be there. Yes, ma'am.
If someone has like white skin,
A white stool.
yeah.
There's a lot. You obviously have an issue, but are we on medication? Are you being treated for something? Is there chemotherapy? Like, there's a lot of questions that you would ask. You couldn't just diagnose on that, but that would definitely be an indicator that something's not functioning correctly.
But I've given my kids antidiarrheals
Mhm.
and caused us to return blood.
Yeah. Oh, so so and whenever that was my question. Are you on some type of medication? Because when you're on medications, your feces can range in colors from white to fluorescent on some antibiotics, like they fluoresce. You're like, what? Yes. So if you're on some type of medication or you're undergoing some type of treatment, it is not uncommon for your feces to be discolored in some way. And even a healthy person, depending on, even if they ate the same diet, the same thing every day, you may see their feces go from like a brownish to a greenish to a brown, like as they're filtering and getting rid of waste. So we expect to see some range of discoloration, but if you're on a medication or undergoing some type of treatment, you don't just look at it and go, oh God, something's wrong, because you're clearly, parents will say, my kid's feces are orange. Well, are they on an antibiotic? Yeah, okay. And what your feces may look like when you're taking that medication may be different, because it's the way that you process it, your metabolic processes.
I have a question. What type of blood are you using when
You're not losing blood when you're menstruating. You're losing the tissue inside of your uterus. So we will actually cover that in chapters 27 and 28. But there's no ready blood that you're not like bleeding internally. It's just tissue that's being sloughed off, and we're going to cover that. We have a whole chapter on it. All right, erythrocyte disorders, anemia being the first one. Now look at this real quick, because people say, oh, I have anemia. What kind of anemia needs to be your next question. Because anemia is like one of those things. It could be a symptom, and it could be a condition. What's causing your anemia? Do you have an issue, and this is leading to anemia, or is anemia your issue? What is it? Okay, so don't ever just say, oh, I'm anemic. Like, have an explanation for it, okay? Ultimately, all anemia means is that your red blood cells don't carry as much oxygen as they need to. So we need to fix it. And we need to figure out why is it that that's the case. So we're going to go through. But usually people with anemia are tired all the time. We see this happening in pregnant moms. As soon as they become pregnant, their diet's not balanced enough to support that baby, so they sleep forever. For like the first 3 or 4 weeks, they're just super tired. And that could be as a result of that. We also see, followed with fatigue, that they have that white blanchish color, like that pallor that we discussed previously. Shortness of breath, chills, so they go from being really cold, but not like feverish chills, but just there's an issue. When we have anemia, we can group them. Is your anemia caused by the fact that you're losing blood because you've been stabbed, which is again the example? Maybe you're just not producing enough red blood cells, or maybe your red blood cells are being destroyed very quickly or prematurely. Hemorrhagic means that you are losing blood. So you have anemia as a result of losing a lot of blood, probably because you got shot or stabbed. Okay, so how do you fix that? Well, you close the wound and you put blood back, right? So that's what you do. You go to the hospital and get it fixed. Hopefully. Chronic, what does chronic mean again? Long term. So you're losing this blood over the series of a long time. 3, 4 months, 12 months. You're constantly losing blood. And this is because you have some type of injury. You might have hemorrhoids. You might have an ulcer or something like that, where you're slowly losing blood. And it shouldn't be something that kind of catches you off surprise. A lot of times these people know this. And there's been a few cases, like perfectly healthy people are like on their deathbed within 24 hours because they had an ulcer and didn't realize it because there was no telltale signs of that ulcer. And So that does happen. I don't want to say you always know, but for the most part, you should probably know. You're losing the blood, you're anemic. How do we fix that? You don't just give someone iron if they're anemic because they keep losing blood. You fix the problem. So it's what the anemia is what we call a secondary problem. You wouldn't have the anemia if you didn't have that primary problem. So if you go into practice medicine and you're the doctor, you're the PA, or you're the nurse practitioner, you want to figure out what is the primary problem. Once you know the primary problem, then all of these other ones just may be as a result of this primary problem. So they come in and you're like, okay, you're anemic, but what's causing your anemia? Right? What if you are just iron deficient? What if your diet is low in iron or B12? Then you just take iron supplements. So iron is what binds inside of hemoglobin and allows it to bind to the oxygen. So when iron levels are low, oxygen levels are low. So all you have to do is increase iron. You don't need a whole bunch of iron. So a small little iron tab is going to be just enough. Okay, taking too much iron will cause constipation and turn your poop into rocks. You really don't want that. Not like rocks, but it'll be. It's pretty rocky. Okay, shark poops is what my kids call it whenever their poop is really hard. They're like, I got shark poops. Oh. You mean like sharp? Because, yeah, but that's what it turns. So don't overdo the iron. You'll cause constipation and rocky poops. All right, so iron supplements there. Pernicious anemia is a condition. It's an autoimmune condition, which means that your body is attacking itself. Can you cure yourself from this? The answer is no. You can only treat it. But ultimately what happens is your stomach lining is kind of eroded away, that mucosa. Not eroded, that's a poor word for it because you're not a physical. Anyway, it's kind of eaten away. You just need to replace the B12, which is what allows you to absorb that iron so that you can get it back in your diet and carry that oxygen the way that you're supposed to. So you have this condition. They have, you can have like a nasal gel where it's putting that B12, because a lot of times it's not going through your digestive system because your stomach lining isn't. So this puts it straight into your bloodstream and allows for that B12 to get there. And it also mentions here if you're a vegetarian, we also see B12 in low levels there. So make sure you take the multivitamin. Renal anemia. Renal tells you we're dealing with what organ? Kidneys. So the kidney is responsible for releasing EPO to make red blood cells. So if you have an issue with your kidneys, you probably have an issue with your red blood cells. Does that make sense? So that's the primary problem right there. And it mentions here that it's usually followed up by a renal disease. So kidney disease is going to follow it. How do you fix an anemia that's caused by a dysfunctional kidney? Well, you're just going to have to give yourself EPO, so synthetic EPO, erythropoietin, which is why I say you can buy that. You don't want to buy it on the black market. You want to go to the doctor and have it prescribed. Don't do anything on the black market. Aplastic anemia. Aplastic anemia is a horrible condition in which all of your cells, all of your formed elements, there's no distinguishing red, white, platelets. It destroys them all prematurely. That's a problem. So normally once you're diagnosed with aplastic anemia, you're in and out of the hospital for a while. The long-term fix of this is going to be a blood transfusion. Let me see. Yeah. We transfusion and then we transplant the actual stem cells. So we're trying to get you healthy stem cells so that you're making the right types of blood cells. Aplastic anemia is not anything to mess with long-term. Okay? And you will probably spend 4, 6, 12 months in the hospital here. Okay? We normally see this happening with kiddos, which is sad, but that's where we see it. Because if you are an adult, normally it doesn't. You've made it past that. Hemolytic anemias. What does lytic tell you? That I'm rupturing it or I'm breaking it, I'm lysing it. So hemolytic means that I'm breaking these red blood cells, and as a result of them being destroyed, they're not able to carry oxygen. Okay? What would cause these red blood cells to lyse? It could be that your hemoglobin is not right. Okay, that's a genetic condition. Incompatible transfusions. So you were given blood that is not compatible with yours, so we have an issue. You're on the way to expiration there. And infections. Maybe you have a viral or a bacterial infection that has caused your red blood cells, depending on what it is, to lyse prematurely. Again, I already mentioned that the hemoglobin issue, that's genetic. Hemoglobin is a protein. Your DNA has the instructions to make hemoglobin. So if your hemoglobin is wrong, your DNA is wrong. It's genetic. All right. We do see this in certain ancestral backgrounds. So like mentions here, Mediterranean ancestry. In this case, your red blood cells are really thin. It's because of the deficiency in that hemoglobin. And they're fragile. They don't carry as much oxygen. Okay, and it says that their subtypes vary from mild to where you can live a pretty okay life to severe where you don't live very long at all. So it really just depends on how it impacts you. A very common anemia in the United States is sickle cell anemia, most prevalent in African Americans that came from an African descent. Because in Africa it's beneficial to have sickle cell anemia because if you have sickle cell anemia, you're immune to malaria. And that is beneficial because there's a whole lot of prevalent, rampant malaria there, and there's not a lot of treatment. So if you have sickle cell disease, you won't die of malaria, but if you have sickle cell disease, you may die prematurely from sickle cell disease. Sickle cell disease is a genetic condition as well. Let me show you kind of what you're looking like. This is what you should be. This is what your red blood cells look like when they release oxygen. Problem with this, remember how I talked about this is perfectly designed to get through your capillaries? When it changes shape like this, it gets stuck. So what happens to the one behind it? There's like a pile up there. So people with sickle cell anemia bruise very easily, and they're tired. Depending on, and there's a range on this as well. Severities can go from being very mild to very severe. I followed a few case studies. One in which a young lady, when she would go to sleep, like because she was in the same position, she would wake up and she'd have a bruise across her body and she couldn't move because there was no oxygen to that tissue. So she would say it's a good day or a bad day depending on how much she could move when she would wake up in the morning. A lot of individuals with full-blown, the severe type of sickle cell anemia will die around 20. And those with who are heterozygote for it will live much longer. But full-blown sickle cell, and you have a severe case, you don't usually live very long. There's a lot of impact. Ma'am? Heterozygote means that you have a dominant and a recessive allele for that, which takes us into biology and Punnett squares. But homozygote would be someone who had full-blown, they were homozygote recessive.
So what's the usual average rate?
If you're a carrier for it, that means that you're a heterozygote. You have a capital and a lowercase. And there's a chance that if you reproduce with someone who's also a carrier, that your child. But if you reproduce with someone who doesn't have it, your child could either be completely normal or also a carrier. It just depends.
I mean, what does it put you on that?
This is either you have it or you don't. Okay, yes, ma'am. This isn't when we could definitely analyze genetics, though, just not right this minute, but for sure we could. Okay, erythrocyte disorders. Polycythemia vera is ultimately you have bone marrow cancer. Cancer means you produce the cells too many too fast, and they don't know how to work right. So it mentions here you have an excess of red blood cells. Problem with too many red blood cells, especially too many immature red blood cells. First of all, they don't know how to carry out oxygen the way that they're supposed to, and your blood is super thick. That's a problem. So it causes premature clotting, issues with all of that. Secondary polycythemia, this is caused by blood doping, so let me tell you how. This is not artificial EPO. What this is athletes start to draw their blood out months before a competition, and then they freeze it. They put it in a cryogenic freezer, and then right before, they will inject themselves back with their own red blood cells. So their red blood cell count shoots up, and it's natural, right? Because it's their own. But because they have so many red blood cells, they can increase the amount of oxygen that they can carry and increase their endurance. So we also now we're not looking for artificial substances now, we're doing a hematocrit test to see how many red blood cells do you have. It's crazy, but it's a thing. Okay? Of course, these athletes, when they put these red blood cells right back into their body, it's good because it's theirs, but what does it do to the viscosity of their blood? It's super, super thick. And that becomes an issue for your heart. Okay? So done with erythrocytes, now we're on to leukocytes. Leukocytes, any questions on erythros? Okay, leukocytes are your white blood cells. So it mentions that this is just a small percentage of your blood volume, especially if you're healthy. In the event that this becomes a larger percentage of your blood volume, that tells us that we have an issue. Okay, so white blood cells are normally like our fighters, and we'll reference that as we get into other chapters as well. So it mentions their function in defense against disease. It says they can leave the capillaries via diapedesis. Diapedesis is not covered in length in this chapter, but diapedesis means that those cells are in your capillaries, and then they stick to the wall of your capillary when there's an injury or an issue, and they leave your blood circulation to search for the problem. Okay? We're going to come back to that in another chapter, but diapedesis, because red blood cells stay in your capillaries. They do not leave your capillaries. But if you have an injury, white blood cells can stick to the sides of your capillaries and squeeze out. And that's diabetes, because they need to go to wherever the injury is. Okay? And we'll see that there. So that's what diapedesis is. It moves through those spaces with amoeboid action and followed up by positive chemotaxis. You had no idea that your white blood cells can smell, but I'll get there later. Leukocytosis is just a situation where your white blood cell count is high. If your white blood cell count is high and it's normally homeostatic, that usually just tells us you probably are getting sick. And that's a normal reaction to getting sick. Now, if you have a sustained high white blood cell count, that tells us that we have a long-term chronic issue, cancers and so on. So, when we look at our leukocytes, we can put them into two groups. Of course, we're going to further subdivide these, so you already knew that was coming. We have granulocytes and agranulocytes. So granulocytes have a grainy appearance when you look at them underneath a microscope. Guess what agranulocytes look like? They don't look like a grainy appearance. Okay? And I'm going to show you on these pictures, you really can't distinguish grainy from not grainy, but still, that's the two major classes. You have 5 types of leukocytes. 5. Neutrophils, eosinophils, and basophils, or eosinophils. You can call them however you want to call them. I call them eosinophils. Neutrophils, eosinophils, basophils are all granulocytes. Lymphocytes and monocytes are agranulocytes. So 5 total types, and this shows you which category they fall into. Here is a mnemonic device, so a memory tool, that helps you remember the order of abundance, which you have the most of. Never let monkeys eat bananas. Neutrophils, lymphocytes, so you have the most neutrophils, then lymphocytes, then monocytes, eosinophils, and basophils. So in decreasing, here's the most abundant, least abundant. There. Okay, here's showing you if you just take, so you did your hematocrit, you did your centrifuge, not your hematocrit. Here's your hematocrit level, your erythrocytes. This buffy layer, the white blood cells, you have your neutrophils, lymphocytes, monocytes, eosinophils, basophils. So never let monkeys eat bananas. That's how it goes there. So granulocytes just means that they have a granular appearance. I already mentioned that the only formed element that doesn't have a nucleus is the red blood cell, or erythrocytes. So these are all going to have nuclei. All of these are phagocytic to some degree. Does anyone remember what phagocytic means? It will engulf something and then decide if it needs to destroy it or not. So phagocytic means that if it finds it, it will engulf it. Okay, so that was a form of endocytosis, so taking it into the cell. So, and we'll look at those jobs a little bit later. So beginning with neutrophils. Neutrophils, I feel like, and we're going to discuss this again, are the martyrs, ultimate martyrs in your body. So when there's an issue, they are there immediately, and they die protecting you. Okay, so that's what they're supposed to do. We often call the neutrophils bacterial slayers, because they just show up and they're like, I'm going to take care of everything, and they die. But they got rid of the first line of defense, that bacteria. Okay? It also mentions that these neutrophils contain hydrolytic enzymes. Hydrolytic enzymes means that when that neutrophil engulfs or phagocytizes something, it immediately destroys it, which is why we call it doesn't ask if this is good or bad, but it immediately destroys it because it's filled with hydrolytic enzymes. That's why we call it a slayer, because it doesn't ask questions. It just engulfs it and destroys it. There will be some that will be antigen-presenting cells that we'll mention later. It mentions also here that the nuclei are 3 to 6 lobes, twice the size of red blood cells, and we're going to look at a picture here in a few slides. Eosinophils. We really only see eosinophils in high levels if you're having an allergic reaction, or you have asthma, you have some type of where histamines are going to be released. And we're going to see histamine again with basophils. So role in allergies and asthma, they modulate the immune response, so they're going to tell your body something's wrong, and then we'll all of a sudden have an inflammatory reaction. Bilobed nuclei, which means it just has 2 lobes. Basophils, again, you're not really going to see these. It mentions that they're functionally similar to mast cells. Mast cells are always going to be present if there is an acute infection, like an allergy or an allergic reaction, or like anaphylactic shock, something like that. Something where your body is in a stress situation. Again, we're not going to see basophils all over. Again, it does mention histamine. When you take an allergy pill, I don't know if you know this, but your allergy pills are antihistamines, right? And so antihistamines are trying to counteract what your body's doing because it's saying things are going wrong. Okay, so an allergy medicine, when you're having an allergic reaction to something, histamine is released, and antihistamine counteracts that in order to get your body to try to come back to homeostasis. Allergic reactions can be mild or severe, and I feel like you might already know that, but some of you may have a small allergy to something where your nose runs and that's it, but some of you may have a severe allergy, meaning you get bit by an ant and it could cause you to die because you're that allergic to it. Okay, so those are the people who carry EpiPens. Here we have a neutrophil, so we have a 3 to 6 lobe nuclei. You can clearly see that there's multiple lobes there. You can see the nucleus there. This is a granulocyte, so it has a granular appearance. Here we have a eosinophil, bilobed nucleus, granular appearance. And now you have your basophil. Here you can kind of see, this is where it's talking about the nucleus, this darker portion here, but it does look granular in its appearance. Okay, but when you compare it to agranulocytes, I feel like it kind of looks. Anyway, it could just be the poor picture quality. So we've talked about neutrophils, eosinophils, basophils. Now we still need to do lymphocytes and monocytes, which are agranulocytes, meaning they lack that granular appearance. The lymphocytes are the ones that people get confused with leukocytes. So leukocytes just means you have a white blood cell. Lymphocytes are a type of leukocyte. When you talk about T cells and B cells, those are lymphocytes, which are a type of leukocyte. So make sure you keep the two segregated. Don't overlap them or misread your question. And we should have a high level of lymphocytes. And this is our overall immunity. We don't technically have an immune system, but we'll discuss that again in a different lecture. We're going to find it in our lymphatic tissue, which is going to be our lymph vessels. We'll study those as well. You have a few that kind of circulate in your blood, but overall, these are what are responsible for making sure that you are protected against things. So we have T cells and B cells. It mentions here that T cells are specific, and these are killers. And when I introduce these to you and we discuss them, I'm going to talk about how aggressive they are. When there's a problem, they go in, they don't ask questions, they destroy. But the only way your T cells will do work is if your B cells or helper T cells communicate that there's a problem. And your B cells are antibodies. And they're kind of everywhere, just trying to make sure that you're good. So we'll talk about those. So B lymphocytes, T lymphocytes, this is just general. I'm going to tell on you if you're bad. B cells are not as aggressive as T cells. T cells touch and kill. But there's a lot to get a T cell. There's a lot needed to get a T cell to actually do what you need it to do. Monocytes, the very last leukocyte, the largest for sure. they are responsible for turning into macrophages. So macro tells you that it's big, and phages are going to engulf things. When monocytes get released somewhere, that's because there's a cleanup or there's an infection. There's something that needs to be maintained. There's an issue, let me come in and clean it. Monocytes are going to take care of that. They're responsible for communicating to the lymphocytes if there needs to be a problem. So let's say, for example, you just get a paper cut. Monocytes are going to go, bacteria is going to get inside of you because there's bacteria over everything. Monocytes are going to go engulf that bacteria. And they're going to, okay, it hasn't really spread around everywhere. There's no reason to launch an immune response. Does that make sense? Now, if you have this really big gash, of course our neutrophils are going to go there, but then our monocytes are going to follow up. And a lot of different types of bacteria will be there. They will engulf the bacteria, and they will carry it back to a lymphocyte and say, look what I found, my king. And the T cells will say, oh, good job. And then they will specifically go out and kill it, or they'll say, not a big deal. Okay, so we'll talk about that when we talk about the T cells. We have a whole chapter on that. Get excited about T cells and B cells. So here is an agranulocyte, which I feel like looks just like a granulocyte. But you judge what you think. Just know the names, okay? The two that are agranular versus the three that are granular. Leukopoiesis, which is the production of leukocytes, so we're just making white blood cells. And white blood cells are made from hematopoietic stem cells. So hemocytoblast, hemo is blood, cyto tells you cell, and blast tells you that it's a baby, it's immature. Okay, so hematopoietic stem cells, you're going to see they all start from the same thing, and then once they commit, they become white blood cells. So here it tells you here, look at how that's titled, please and thank you. I mean, you don't. Hematopoietic stem cell, so the same thing we had for red blood cells. Then it says hemocytoblast. So it's giving you interchangeable terms for this stem cell for all blood. Then, if it's going to become a white blood cell, a leukocyte, it will either become a myeloid stem cell or a lymphoid stem cell. The lymphoid are going to be the B cells and the T cells, the lymphocytes. The myeloids are going to be your neutrophils, the eosinophils, basophils. Okay? So you can see that here in monocytes as well. So you can see the distinguishing between the types of leukocytes. So lymphocytes are a type of leukocyte. Leukocytes are white blood cells. Questions right now? All right? Leukopenia, anything tells you that it's low. So leukopenia would mean that you have a low level of white blood cells. Why would that be a problem? You can't fight off infections. So leukopenia is an issue. A lot of times, if you may have low levels of white blood cells because you have a disease or a condition, or you might be being treated for one. So in the event that you're being treated for a condition that's causing leukopenia, we're probably going to keep you in the hospital. Why do we want to keep you in the hospital if we're going to lower your white blood cell count? Yeah, we want to make sure that we react to anything that you're infected with, because you're more likely to become infected. You're already compromised, and now your white blood cells are low. Leukemias, this is where you're overproducing the leukocytes. So overproduction of white blood cells. Now, the type of leukemia is distinguished by the name. So which leukocyte is being overproduced, that's how you know what you're dealing with. But if it says leukemia, it's an overproduction of some white blood cell.
And the different types is what type they're attacking.
Right, which white blood cell is being overproduced? Yes, ma'am. Okay, so anytime it's a cancer, we already know that we're making too many and they're immature, so they're not carrying out their job. Named according to the white blood cell that they're using. Anything that's myeloid leukemia is going to be neutrophils, basophils, eosinophils, or monocytes. If it's lymphoid or lymphocytic leukemia, then that's going to be your T cells, B cells. Remember, you have 5 types of leukocytes. Acute leukemia is usually what we see in kiddos. If we see a leukemia show up in an adult, I'm not saying it's rare because it's becoming more and more common, but it's usually because there was some issue that kind of led to it that maybe they didn't know about. So it's chronic, meaning that it's taken place over a really long time. Like you didn't just develop it today and get diagnosed today. Like if you're an adult and diagnosed with leukemia, likelihood is that something was triggered over a year ago to get you to that state. Okay? Ultimately, whenever you have any type of leukemia, it becomes an issue because the cells that are making your white blood cells are your hematopoietic stem cells. So all the other blood cells, because you have so many white blood cells, are crowded out. So if red blood cells are crowded out, we have anemia. We'll have issues with that. We'll have clotting issues, which would be my platelets are being kind of crowded out. We already know that cancers are immature cells. Usually people who have leukemias die because of an infection or an injury that they are not able to heal. Okay, which is also why they stay in the hospital. Treatments, we have radiation, you have any type of cancer treatment, and then ultimately long-term, if we can get you to survive that long, we're going to do stem cell transplants so that we can get you healthy stem cells so that you make the right type of leukocytes. Infectious mononucleosis. This is mono. People say, I don't have mono. Technically, it's Epstein-Barr syndrome, or the Epstein-Barr virus is what causes it, and it's named after someone, which is why it's capitalized. Because it's a virus, all it is monocytes. You have a whole bunch of monocytes. It's viral, so can you take a medication for it? No. You cannot. Anything that's viral, you cannot treat with medication. No viruses can be treated with medication. So you get mono, you go to the doctor, they'll take blood to diagnose you with mono, and then they'll say, hey, you have mono, your body will fix itself in about 6 to 8 weeks. So there's no medicine or anything like that for mono. A lot of times you just have a kid or an adult who's crazy tired and really has no reason to be crazy tired. Like some of you work a lot and you do a lot of things, so you are justifiably tired. But if you have a kindergartner falling asleep at playtime or craft time, then that's an issue. So you have that kiddo checked out. It is contagious. That's why it's called the kissing disease, but you don't have to kiss to get it.
That's what I was just trying to think of.
You process it. People always say, oh my goodness. So if you do have mono or someone in your home has mono, you use separate straws, you don't share drinks.
But you didn't necessarily
kiss somebody, yeah.
Wow.
Oh, these two people have mono. No, it doesn't have to be like that. All right, any questions on leukocytes? Good deal. Platelets, the last one, which we'll end up calling thrombocytes. All platelets is they started off as this really, really large cell, a megakaryocyte. So mega tells us big, karyo tells us nucleus, and cyte tells us cell. So it has this really, really big nucleus, and then it just fragments into all these little cellular fragments. And I'll show you the progression picture there. We expect platelet levels to fluctuate, let's talk about what they do and then talk about issues that go along with that. So the purpose of a platelet is to create a plug in the event that a vessel is compromised. To create a platelet plug. That's what they do. They create a clot, which is like a scab, and it allows that tissue to heal so that you don't lose blood. That's the goal. Most of the time, for those of you who are healthy, because you do have platelets, you don't have these clots all the time because you're releasing nitric oxide. Nitric oxide is saying don't clot, don't clot, don't clot. So you're not clotting. Okay? They naturally want to clot. That's their job. So they have to be told to not clot so that they don't cause these crazy random blood clots for you. We do know that these cells will age very quickly. They will be gone with about 2 weeks, and you'll have made others to replace them. It mentions that they're regulated by the hormone thrombopoietin, because thrombo is going to be anything platelets, leuko is white blood cell, erythro is red. So thrombopoiesis, thrombopoietin, you're going to see that same pattern as we saw in the previous two. Again, they're derived from a megakaryoblast, which will eventually become a megakaryocyte cell. Sorry, let me show you what we're looking at. So we have a really big cell with a really big nucleus, and you can see that, and then it just fragments. So it's not just one cell, it was one really large cell that fragmented, and now I have a bunch of platelets here that can be used for clotting purposes. Okay, here is all the formed elements. So we have the erythrocytes, the 5 types of leukocytes, and then the platelets, and it gives you distinguishing characteristics and statistics on each one. Hemostasis. Hemo tells us it's blood. Stasis is a balance. Hemostasis is the process of making sure that you're keeping your blood volume constant. The only issue we would have with that is if there was some type of compromise. So like you had a cut or a gunshot or an ulcer or something like that. Hemostasis is a series of reactions that stops you from bleeding, and this requires clotting factors. Again, the majority of us are healthy, so you clot perfectly fine. We are going to talk about hemophilia briefly in just a moment, which means that you don't clot correctly. But there are 3 steps to your clotting process. A vascular spasm, which tells us that that blood vessel has been ruptured, so it kind of like flips out. So it's been ruptured, so it begins to spaz, like contractions. Okay? Then we form a platelet plug, which is a temporary scab, and then we coagulate. We have a whole blood clot. Then we'll do clot retraction. We'll pull those vessels together, and I'll show you what I'm looking at. Here, we're just hanging out, doing regular things, and all of a sudden this blood vessel is ruptured. So you can see that there's erythrocytes there, and that there's a platelet depicted. But once there's a rupture or some type of compromise, the platelet count increases. And their job is to come and form a platelet plug to keep you from losing blood. This can take place, depending on the size of the cut, in a couple seconds. It may take a couple minutes. If it's a huge laceration, you're not going to be able to create a platelet plug to close that. Does that make sense? So it's just going to depend on how bad the damage is. Once you form that platelet plug, we'll form a scab. And you form scabs on the outside, so those are ones I'm going to reference because you can see those. But you also form scabs on the inside too. And these scabs, the purpose of a scab, this right here is a protein called fibrin. And as that scab disappears, so if this was the vessel before it was ruptured, once it's ruptured, it separates. You form that scab. As that scab disappears, it pulls the 2 sides of the vessel back together. So it like slowly brings them back so that they can heal, and there's no need for that clot anymore. There's no need for that scab. Your scabs will heal from the outside in, which if some of you are scab pickers or you know people who pick scabs, it's the edge of the scab that comes off first. And then if you keep peeling that scab, when the middle's not ready, it exposes a wound still. And then the scab will have to come back. So just so you can kind of relate this process that we're discussing here. Here's what a scab looks like up close. We have this fibrin mesh, okay? So we have a bunch of protein here trying to hold this stuff together. And then red blood cells get caught in it because that's what they're doing. It's clotting, okay? So that's what a scab looks like up close. Clot retraction. So I've formed my scab. Now I need to bring my 2 sides back together, my 2 loose ends together. So it mentions that it stabilizes that clot within about 30 to 60 minutes. And again, this is depending on where it is and how severe it is. It begins to start the process. That process may take a couple hours and it may take weeks, depending on how big the issue was. But ultimately, clot retraction, we're drawing the ruptured blood vessels back together, those edges back together to form a smooth, nice new red blood vessel. It mentions here that the vessel repair is where the vessel heals. We've used the term endothelial before, but thelial tells you that it's what? It's exposed to the environment. Endothelial tells you that it's inside and exposed to the environment. Inside of your red blood vessels, inside of your veins, it's all lined with epithelial tissue. Why is your vessels, why are your vessels lined with epithelial tissue? Oxygen comes from the environment, your nutrients from your food come from the environment. Anything in your body that is exposed to the environment is lined in epithelial tissue. So endothelial is what we call the epithelial tissue that's inside of our vessels and our heart. Okay? So it does heal very quickly because we know epithelial tissue has a high regenerative capacity. And this is what it's mentioning here. Fibrinolysis is the breakdown of the actual scab. Okay, because the scab was made from the protein fibrin. So fibrinolysis is the lysing or breaking of fibrin. So we're breaking down that scab. We're getting rid of that scab. The name of the enzyme that does that is plasmin. Plasmin is the enzyme that leads fibrinolysis. Factors that may cause you to clot undesirably. So when you shouldn't be. Well, first of all, for you and I who don't clot undesirably, we're releasing nitric oxide. Our endothelial tissue, our layer on inside is saying, don't clot, don't clot, don't clot. You don't need to, I'm not damaged. So we have that already happening. And it mentions that the endothelial lining of our blood vessels prevents that clinging. We have antithrombic substances. Thrombic tells you platelets. Anti would mean anti-platelets. So we're trying to detract or deter platelets whenever they're not needed. Okay. Vitamin E also acts as an anticoagulant. Multivitamin, you're good. Thromboembolic disorders. So this is what's going to get us into the undesirable clotting. Thrombo tells you that we have a clot that showed up for no reason. Embolic means that it's an embolus, which it means it's broken away, and it's now floating. And these, I believe these terms are defined in a slide or so. This means you're making clots when you don't need to be making clots. That's an issue. If you're not damaged and you're making clots, there's no reason to have that. When those clots break off, they get stuck in smaller vessels and they can cause embolisms. So a cerebral embolism, one in your head, or one pulmonary in your lungs, and then you die. Okay? Bleeding disorders means that you bleed too much, which means you don't clot, so there are hemophilias. Disseminated intravascular coagulation. This is where you have a clotting issue that causes a bleeding issue. So you have both. And that kind of seems weird, where you overclot and you overbleed. Those are both, that's an issue. So a thrombus is, by definition, a blood clot that forms when there's no need for a blood clot to form. So it just decides to clot, and now you have a blood clot. That's a thrombus. A thrombus escalates to an embolus when it breaks off of that vessel and begins to float in your blood. That's an issue. An embolus is an issue. An embolism is when that embolus gets stuck somewhere. And it can technically get stuck anywhere. Some areas are less lethal than others. So if it gets stuck in your brain, that's a big deal. If it gets stuck in your lungs, that's a big deal. If it gets stuck in your heart, that's a big deal. Okay? And risk factors, atherosclerosis, meaning that you have a buildup of cholesterol or issues with cholesterol deposition in your vessels. Inflammation, so you could have some type of infection or injury. Slow flowing blood or blood stasis from immobility, meaning that you don't do what? Move. You just stay in the same spot. We see this a lot in our geriatric patients, our elderly.
How do you bleed like through your skin without a rupture?
Right, so it causes bruising, and there's no need for the bruise.
I mean, like to literally
Yes.
through.
Right, like you can
outside.
see. Well, if it's bleeding within your skin, so you're saying that there's some type of cut. If there's a cut, it's going to continue
Is it
to
like a no cut? Can you bleed?
through your skin.
I mean,
I
think they said she had maybe that. That's what she had, and it caused like a blood vessel to rupture or something. And then
Yeah, so when a blood, like if your skin is intact, the blood won't leave. Your lymphatics will pull it up, but you can have a nasty looking bruise or that blood is beginning to, because that's all a bruise
like
is.
something like that happened to my mom, but it was just she was just bleeding with no cut or nothing.
I don't know. I would have to look at that in that case study, yeah. Because yes.
Blood thinners.
Like that's what there would have to be some type of. So anticoagulant drugs. If you have a clotting issue, aspirin, and probably many of you have already seen those commercials. I know that when I was little, my pap used to have to take aspirin all the time as a blood thinner. They say take a Bayer a day and your heart is good. So I don't, but that's what my grandpa did. I take multivitamins every day.
I have a question. Why is aspirin not good for people with asthma?
With asthma, it depends on the type of asthma and what it triggers it. So it's not just saying that asthma in general. So that's a great question, but those are very specific. Like it's hard to make those general comments on all of that. Heparin is a blood thinner, so that's an anticoagulant. Warfarin, same thing. Dabigatran, same thing. And all of these work to keep you from clotting. Okay, so they're anticoagulants, anticoagulant drugs. Bleeding disorders. Thrombocytopenia. Thrombo tells you platelets, cyto cells, penia tells you that they're low. If you have a low amount of platelets, that tells you that you're not going to clot, which means that you're going to probably do what if you're injured? Bleed out. Okay, that's going to lead us to hemophilia. Petechia. Petechia, some of you have heard that term before. You can often see it in people's eyes when they're having issues. It kind of looks like, if you've ever seen like a shotgun, what is that called? That just shoots out? I forget what it's called. But anyways, a spray of a shotgun, that's what petechia looks like, especially if somebody's been strangled or oxygen levels are low. You can definitely see petechia, and you'll say that individual's petechic. We can tell if there's a clotting issue there was some bleeding, some rupturing, whether it was self-induced or somebody else did it, or you're perhaps just sick. So there could be a lot of different reasons why that would take place. Ultimately, if you're having a clotting disorder, we need to either figure out what's causing that, what's causing your platelet levels to be low and fix it, or we need to transfuse the cells that allow you to make those platelets. If you have an impaired liver function, you might also have a bleeding disorder. So any liver disease has a large impact. So it's not just going to have one sign or symptom. It's going to affect a lot of different things. So this individual is probably going to be jaundice as well, kind of a yellowish color. But you can see here that it continues to talk about that. It talks about vitamin K. Individuals with hepatitis, that's a liver issue, can have bleeding disorders as a result of that. So it's a secondary condition. Hemophilia is you're missing a specific protein that allows for clotting. A protein factor, a clotting factor. So there's 12 factors. If you're missing factor 7, and we know that that's the one that you're missing, we can just give you factor 7 and treat you with hemophilia. This, in the old days, would cause people to die. They would get a paper cut and it would just bleed for hours. A paper cut wouldn't kill them, but a larger cut would have, just so that we're on the same page. You didn't just paper cut somebody and then die. Disseminated intravascular coagulation. This is the one I was talking about where you have a combination of it. This is you overclot, but you overbleed. So ultimately you're clotting, and because it's so clotted, it causes the blood vessel to rupture. And because the blood vessel ruptures, you bleed a lot. And you can't stop the clotting there. So there was a clot that led to overbleeding, and it continues to do this. And ultimately, it can lead to death. You're going to have a blood transfusion if you're going to stay alive. So you notice that when we talk about the big conditions on all of these, blood transfusions, stem cell transplants, because it impacts all the blood cell lines. What is a transfusion? A transfusion is where you're taking blood and you're putting it into someone, and there's different types of transfusions. If you just take red blood cells, you can pack red blood cells and give them. Sometimes you give whole blood, sometimes you just give some of the elements of the blood, like platelets or red blood cells. And those of you who have donated before may have had your blood separated out, especially if you're O positive, they're going to take blood from you all the time. If you give somebody the wrong type of blood, it can and usually is fatal if nobody catches it. When you're in a blood transfusion, you should be in ICU. So when we talk about the blood groups, humans have ABO for their blood group or blood type, and this determines which type of blood that they can interact with. This is genetically predetermined by the parents, and the baby is a result of the parents. Let me kind of show you what we're dealing with here. Again, the majority of us are ABO. If you have a different type of lineage or ancestry, where you've kind of stayed in an island for hundreds of years, you have a different type of blood. Like we have Mediterranean blood types like Duffy cells. We have all kinds of that, just not the vast majority of us. So if you've stayed in an isolated area, you can definitely have that. Okay? We have type A, type B, type AB, and type O blood. And then we have the positive and negative, the RH factors that go along with that. Ultimately, your blood type is determined by what type of antigen you have, and that is genetically predetermined. You have that at birth, but you have the right antibodies and antigens by the time in full circulation. Around 2, for sure, by the time you're 10, but you should already have a pretty good set on what type of blood you have from birth. But the full levels of adult circulating blood cells by the time you're 8 to 10. I like this diagram because it gives you a visual depiction of the difference between A, AB, O, and B blood. If you have type AB blood, here's your red blood cell. You have markers on your blood that says, hey, I'm A, and hey, I'm B. So it's marking itself as being both A and B. As a result, there are no antibodies floating outside of your cells if you have AB blood. Because there are no antibodies in the blood, you can receive any type of blood because you won't fight anything because you have no antibodies for the blood. So people with type AB blood can receive any type of blood. They're considered the universal recipient because they don't have any antibodies hanging out to attack the wrong type of blood. People who have B blood have red blood cells that have little markers on them called antigens that say, hey, I'm B blood. I'm B blood. The antibodies present in B blood are anti-A antibodies. Anti-A means if you put A blood in someone who has B, it immediately attacks them. So anti-A means it's against A. People with A blood have antigens that say, hey, I'm A, and their antibodies are anti-B. So as long as A is there, they're good, but if you put B there, it immediately attacks B. People with O blood have no markers. People with O blood have no markers. O is a recessive blood type. But they have both antibodies, A and B, anti-A, anti-B. So O blood can be given to anybody. So again, if you have type O blood, you're asked all the time to make donations. I literally get a call daily, a voicemail daily, and an email weekly that says, hey, we need you to give blood.
But O can only take O.
O can only take O because it's completely recessive. If you give it a dominant type of blood, you will kill them. So O is completely recessive. Now, so O is the universal donor, AB is the universal recipient. Type A can get A blood or O blood. Type B can get B or O blood. Type AB can get any type of blood. Type O can only get O blood. And it tells you all this in this chart. Yes, ma'am.
You said that O is the most recessive?
No,
So
it is recessive.
which one is the most, is there a dominant?
They're what we call codominant. So all of these are dominant. It doesn't, if you have A, that's dominant. If you have B, that's dominant. If you have AB, it's dominant. O is just the recessive one. Good question. Okay, now whether it's positive or negative is determined by what we call the RH factor. RH comes from Reese's, the Reese's monkeys, because that's how we knew this. Not like Reese's Pieces, like RH. It's a type of monkey. Anyways, this is a D antigen. You don't have to know many details about this for anatomy and physiology at where we're at right now, but you do need to know the implications of a pregnant mother with a baby, and they don't have the same RH number or RH sign. So, for example, if you are a mother with negative blood and your baby has positive blood, when the mother does a vaginal delivery, there's going to be some baby that kind of mixes in with mom, and mom, the first time, there's no issue reaction-wise, but the mom becomes sensitized to that. if that mom, and this only happens with mothers with negative blood, and the baby has to be positive. So it's negative, negative, there's not an issue. If it's positive, positive, not an issue. It's only an issue if the mother's negative and the baby's positive. The second time that that mother goes in to deliver, she will either die or the baby will die. Because when you first get a sickness, it takes you a while to build up an immunity to it. But the second time, and we'll discuss this in a different chapter, you react much quicker. The first time mom gives birth, she becomes exposed to it, so her body builds up a rejection to it. And so the second time she gives birth, either the baby dies or mama dies. In the event that you are negative and you're a mama, so it mentions it's the transfusion reaction. This situation is called erythroblastosis fetalis, which is where the mother is negative and the baby is positive. We sensitize you, or to prevent sensitization using a RhoGAM injection. So if you are a negative woman and you've already had a baby, we don't actually type your baby if you have negative blood. Now we don't. If you have negative blood, we're going to treat you like your baby is positive. Does that make sense? Because the only way you're going to know what type of blood your baby has is if you go in and type them. And that can have a lot of risks associated with it. So if you're a negative mama, at 7 months we give you an injection, and right before you go in to deliver, we give you an injection to help prevent that sensitization so that you can continue to have babies and there's no rejection or transfusion reaction.
And then you do that
Each time. And we normally see, the only times we really see any fatalities now with that are people with ethnic or religious issues that say, hey, no, I can't do that, and they won't take transfusions, or people who go in and haven't been treated prenatally. So you didn't go to the doctor while you were pregnant, and they didn't, you come in and you're already in labor, and you're about to have this baby, and you've not been treated at all. So it's not, it is still a thing, but it's not nearly as common as it used to be. So when mom is negative and baby is positive. Okay, transfusion reactions. If you get the wrong type of blood, immediately what begins to happen is it coagulates. So it starts to create clots, and it coagulates everywhere. So it's supposed to be running, and it starts to clot up, and then it gets stuck in your vessels, and then it causes expiration. What we can, initially, it doesn't happen immediately, but initially we can see signs of these transfusion reactions. If you go in for an emergency blood transfusion, we're going to monitor you, and we don't have time to blood test. We're going to try to get you O. But there still can be issues with that. When you have a transfusion, you're going to be kept in ICU. We're looking for your O2 levels to stay high. We're looking for your blood pressure to stay consistent, not drop or anything like that. When we start to see these signs, like low O2, low blood pressure, that tells us that perhaps you're rejecting that blood. If you're rejecting that blood, we need to reverse it immediately. We don't need to wait 4 or 5 days because you're going to be dead. So transfusion reactions can be reversed as long as we've monitored you and we can tell, hey, something's not right. So we can see that your oxygen levels are lower, your blood pressure is dropping. This can ultimately lead to kidney failure, but we try to monitor that ahead of time. What we can do is, if we know, and here are your general symptoms, but of course you should be hooked up to a monitor, an ox monitor. Fever, chills, low blood pressure, rapid heart rate, nausea, vomiting. We pump you full of saline because we don't want to put another blood in you right then, so we're going to try to dilute the situation. Okay? And we're going to cycle that blood out of you slowly. You'll stay in ICU for a few days, if not a week or so. But and that's only if you're having a transfusion reaction, like you're rejecting that blood. Type O is universal donor. AB is universal recipient. Autologous transfusions are where you pre-donate your own blood and you say, I just want you to freeze this in case I need it. And so it's stored for you to use it if you need it. And people do that. We try to blood type and cross-match people before we give them a transfusion in order to prevent any type of transfusion reaction. We try to take the bloods and mix them. If we know that you're going to donate an organ, we do the same thing. So if any of you have gone through the organ donation process, it's not like, oh, you've got a kidney and I need a kidney. It's a long, drawn-out process for that. Here is the blood typing that is done. So if you have type AB blood, if we put you in just A, you will see it coagulate because A blood has B blood. I should be pointing here. A blood has anti-B antibodies, so we'll see coagulation there. B blood has anti-A antibodies, so it's coagulating there. I can tell you here that this is coagulating with anti-B, or anti-A, so that's telling me that this is B blood. That's why it's not, why am I not, I'm looking at this and saying that, sorry. Anti-A means that I have type B blood. So B makes anti-A, type A makes anti-B. We want it to look like this. This means that it's running, it's not coagulating. This is the clotting and the coagulation of red blood cells, which leads to damage. So because it's not clotting with anti-B, I know that this is A blood. This is clotting with anti-A, so this has to be B blood, right here. Okay, so I put it with something with anti-B, I put some B blood in there, or anti-A, so that's got to be B blood. It's wrong. AB blood will do that when you mix those two. Here, right here, it's not clotting with anti-A, so this has to be B blood. This is clotting with anti-B, so this has to be A blood that we tested it with. So the good part of this, what we're looking for is this. If I see that, I know that that's bad. O blood is not going to clot at all, no matter what you put with it, anti-A or anti-B. So I can mix it with A or B blood, and I'll see nothing there. Okay, so when you're studying this, and you can see, obviously, even as I'm reading it, I still get confused. Anti-A will be present for B blood. B blood fights off A, but B has B antigens. So listen and read the question. The antigen is what says this is what I am. The antibody is what it fights. Okay? If it's got anti-B antibodies, it has A antigens. So just read it. Make sure you read those notes, please. And the question when you see it. Again, restoring blood volume. If you lose a lot of blood, we want to naturally put blood in you, but we don't know what blood we can put in you unless we have a lot of O, and then we'll pump you with O. But we would put saline or some type of plasma expander, which would expand the plasma content of what you already have, just to get us by until we can figure out what type of blood we need to transfuse to you. When we do blood tests, you do a hematocrit test to test red blood cell counts, so that also allows us to test for anemia. Blood glucose test tells us how much sugar is in your blood. Do that for diabetes. We look at your blood under a microscope if we're trying to diagnose some type of issue with red blood cells, or maybe even a high white blood cell count, so tell us that you might be sick. Continue with that. Differential white blood counts tell us what type of sickness you have, because remember you have 5 types of white blood cells. Do a SMAC test. This does a whole holistic blood profile. Complete blood count checks each of your formed elements, your hematocrit and your hemoglobin levels. And many of you, when you have your yearly physical, and a lot of your companies pay for this, they test everything. They draw your blood and they run every single test on it. Not like STDs or pregnancy tests, but like healthy tests. So there's a lot of confusion there as well. Developmental aspects, you're developing blood, or you're making blood early on. When you're initially conceived, it's a yolk sac. Once it starts to develop, once that yolk is gone, you're making your own, and we'll talk about that in chapters 27, 28. The fetus forms hemoglobin, but it's fetal hemoglobin, which is much stronger than adult hemoglobin, or normal hemoglobin. Fetal hemoglobin will steal oxygen from the mother. So that's why a lot of mamas are tired. So I talked about anemia previously, and I said a lot of moms, early on that they're tired and they're really laying down their baby's taking their oxygen. Your body will starve you to make sure the baby's taken care of. If your oxygen levels are low, the baby's oxygen levels are low. Okay, so your body needs to rest. You need to make sure that you're getting your multivitamin, in this case prenatal vitamin, because you're pregnant. Blood diseases, we see once you get older, and it's talking about aging here. Chronic leukemias, again, chronic means you've had it for a long time. Anemias, usually because of disorders, eating maybe, clotting disorders. Usually you have these problems because, though these are secondary problems because you have some primary disease. And you need to treat the primary problem before you can treat the secondary issue. All right, let's take a break, and we'll come back to talk about the heart.